upgrade, and this is our company here. Startup company, as I said, we are based in New York, but we're not a startup in healthcare industry. With a great, big, and strongest team we have. And we're very strong in a synergistic combination of generic FDA-approved drugs, and we're creating novel pharmaceutical composition and new novel application for them. And we also, Dr. Farber did mention about it, developing novel pharmaceutical compositions as well. And that's our little tip of the iceberg. We have a key advisors and many more support team. Well, antimicrobial resistance, it's all about bacteria. And this is in blue color here, just one of the statistical menu, what's going on currently. Cancer taking over as far as death situation in our human lives, and rest of the doing their own aggressive job, like viral infections here, some diabetes involved. And we're seeing this nice, unfortunate growing quite extremely. It's already reaching at the cancer level, and it's gonna go more. It's called AMR, antimicrobial resistance. That's going by 2050, which is time flies fast. Unfortunately for this part, we're gonna expect 10 million and more death. Just only one factor besides the other illnesses. And this is really crucial right now, and you will understand why I said that. This is a statistic from CDC, and just I put down some points here on their website. They have a 5 or 6 pages of these monsters, or depends how we look at it. You will understand this later. It's not really monsters as they look. This bacteria depends how you work with them or how you treat them, and I will explain to you later. But meanwhile, this is the bacteria, gram-negatives mainly. That's the topic for this presentation, who are really causing the problems, death. It's different names here, Pseudomonas, Acinetobacter, Bacters, and so on and so on. They're all resistant, Klebsiella, they're all resistant bugs, and it's growing and growing. There's a problem. When you have an enemy, you have to have a weapons. You have to have some defense. You have to have a structure. And yes, we did. We have a bacteria, and we come up with antibacterial weapons against the enemies at some point. And guess what? From 19 to 87, we had a huge development, pharmaceutical grow on the antibacterial field, the antibiotics. Different class, different groups, they were pretty working good, great, helping out. Survival rate was great for the people with infections, all kind of level. And suddenly, and then we see the trend of decreasing of amount antibiotics, not because the amount was decreased, amount of, I'm trying to say novel antibiotics, because what's happened? I guess either we didn't start doing pretty good, or enemy became smarter. I guess number 2, enemy became smarter. It's a bacteria become more functional, and they created defense mechanisms against our weapons, our antibiotics. And the pharmaceutical companies still coming up with new medications. Even actually, I would say this tube should be a little higher, but the problem is they don't last long. They use them for 6 months up to a year, sometimes 2, and then bacteria creates resistant phenomenon, resistant factors to it. So what's going on currently? They're trying to come up. I just put down here a few options what are there on the market. Pharmaceutical market, they're trying to come up with antibodies, probiotics, vaccines against bacteria, not just virus, for the bacteria, some immune stimulations to even the fighting with one of the defense mechanisms as a biofilm peptide. They trying to come up with options to potentiate current antibiotics and conquer this multi-drug, MDR stands for multi-drug resistance gram-negative bacteria. And here we come, TRIZ as the one of the leading part in our, it's actually one of the leading part in our company. And this is where company who are only one of the first company which applying TRIZ in pharmaceutical research and development projects and in the health industry. You know, for a fact, TRIZ was utilized a lot in technology, product development, engineering, computer industry, process management. But in the medical field, we are the one of the first pioneers in that matter. And our study is discovering R&D, research and development fields. As Dr. Farber also mentioned, it's based on computer mathematical modeling and with applying TRIZ principle, of course. So current pharmaceutical industry and always been, they come up with options as traditional approach. It's trial and errors. They're studying, they're trying, errors, they're trying again with different options, which is great. It was worked for a while, but it takes much longer, takes more timing, more expensive. And unfortunately, as always, I say, disease doesn't wait. As we sleep or something going, disease doesn't. The problem's still there. Bacteria haven't slept. They finding new options how to resist our treatments. So our approach involving mathematical modeling and TRIZ principles gives a great advantage to this industry. And this is just quick principles, and that's what they use industries, and that's great TRIZ principles in Samsung, General Motors, NASA, and other companies. So what is all about this polymyxin? What is so many, why would we spend time and present this? I would think this third point sentence here should be first. NIH, National Institute of Health, named polymyxin as recent years as a last resort therapy. There's nothing else. It's a last resort against gram-negative, multi-drug resistant, and now we have another bacteria who are extremely drug-resistant pathogens. They don't respond to anything. They just resistant to all. And they call it last resort. But what's happened to polymyxin? Guess what? It came up to the market. It was actually discovered in 1947. And 3 years later, it came up to the market, and they started using it. And then one of the unfortunate great medications, it's great on the battlefield against bacteria, but unfortunately, it causing damages to the host body, as called nephrotoxicity, kidney damages, causing kidney failure, kidney don't function well, and so on, and could cause at some point more problems than help. And that's polymyxin was placed standby. They used it over the years, but hasn't been used as only used for these certain conditions. But nowadays, they coming back to this medication because there's all other medications were great medications, but hasn't been function well because of resistance. And polymyxin still on the market and used for those cases. What's happened? Why it's causing that problem? Apparently, there was found out that polymyxin loves that megalin. It's a natural, our receptors sits on the kidneys, and job of megalin to actually put, it's working on the kidney and proximal tubules, and it's causing reabsorption or bringing back the proteins back to our body. That's what one of the function it does, megalin and cubilin, endocytic receptors. And when polymyxin giving to the patient to do the job against bacteria, he attracts to megalin for certain reason because of receptors. Receptors, it's called like a ball and the glove. They matching to that structure of that specific receptor. And when it gets there, it's interfere with our kidney function, was most important thing, and we end up with nephrotoxicity and kidney failure. And that's the problem. So main effect, it's great, but unfortunately at the same time, it causing already debilitated bodies, patients having issues already from disease and another problem. Well, they realized that, and they tried all kind of approaches, past strategies. They tried to give intravenous, I mean, it's given intravenously, but they gave the different small doses, different timing, twice a day, once a day, every 8 hours, every 6 hours, depends from condition. It didn't work. They tried analyze delivery methods, intramuscular injection, subcutaneous, intravenous. It still didn't work. This is based on the studies and protocols they use in the hospital field. They even tried to do the purification method for polymyxin, and still didn't work as good as they expected. They tried them in hemoperfusion. This is like dialyzing the blood, and still didn't have any great results as they would like to help to the patient. Coming back to TRIZ, that's our, I would say, alma mater, what we stand on, and TRIZ and mathematical modeling. Well, before you start for the resolution, you have to have a contradictions. So from one side, in order to effectively treat infectious disease and save lives, we should use polymyxin. That's only choice, last resort we have based on NIH, unfortunately, sorry. From another hand, polymyxin causes kidney toxicity, kidney damage, and which prevents polymyxin from full therapeutic dose. What's happening is you don't use, let's say if necessary, 300 milligram, 500 milligram, you give them 150, smaller amount to prevent the kidney damage. It's not enough to conquer the problem. And since polymyxin underutilized, that's another problem. You underutilize, you're using for the patient, you're getting treatment, ongoing therapy, and it's not enough to beat or to bring to the death of the bacteria. Those bacteria which survives, they become smarter, we can say that, or they adjust to the antibiotic, and we getting a resistant. Some bacteria become resistant to polymyxin now, and it brings it to the high risk to lose our last resort therapeutics, which we don't have any other right now antibiotics which could substitute it currently. So some of the examples, I'll tell you why we come up with this mono, bi, and poly hull, and we're talking about stability. When you have just a monohull on a sailboat, or two or three trimaran, catamaran, we always say which one are stronger, which one are steady, which one are powerful. I guess more you have support, or it makes you more steady and more powerful when you maneuver, when you move as well, and less chance to sink. And another, our strongest point is synergistic combination. If you remember, we mentioned one of our division. It's one of the examples from our daily life, combination steel and concrete, and we have a great, strong, powerful for years, ages, like bridges or constructions. So it's just example of synergy and prism of TRIZ. So we have traditional approach. We have a problem. We trying to figure it out what to do with this problem. And we have trying traditional way, as we call trial and errors in the regular pharmaceutical company, and still we cannot oversee that oversee that problem, or resolve it. And then we decided getting backwards, and this is we're using a prism of TRIZ when we have, and 40 principles of trends, technical evolution, and database. When we use world's conceptual problems, conceptual solutions through abstractions, and utilizing by triggering specific applications, we're coming up to solution. So TRIZ principles. What do we utilize in this concept, in this specific scenario? The principle of imposition. We separate the interfering part from the object, which is property of nephrotoxicity. On the contrary, select the only necessary part, the desired property, antimicrobial activity of the polymyxin. We want to deal with nephrotoxicity of that medication, and at the same time, we have to maintain powerful effect of antimicrobial activity. That's what basically makes this, that's what we need for the functionality. The principle of local quality, to go from one object structure or external environment, external influence to a non-uniform, and different parts of the object should perform different functions. Each part of the object must be in the condition most favorable for it to work. So we're utilizing different objects, and we want to bring them to the point, to the solution, to the solve the problem, for the action. Imposition, again, principles, research, and discoveries now. So imposition was, when we use that principle, imposition was achieved by removing interference with polymyxin nephrotoxicity. Local quality, we confirmed that our knowledge of microbiota should be focused on megalin receptors. So we have polymyxin nephrotoxicity, megalin receptors, we mentioned before. That's what the main our relationship and targets. And then principle of pre-plant pillow. I love this principle, which tell us to compensate for the relatively low liability of the facility by prepared emergency. It led us to investigate thousands FDA-approved generic drugs you've seen from Dr. Farber's presentation, as he mentioned about thousands, and substances to serve as nephroprotectors. And this is one of the tools we use in our laboratory, our team. It's what they call it a docking design. When you have a target, we have a ligand, we trying to match, we have a megalin receptors and substances generic. We matching up, see which one will match and cause no damage to our target, but actually will block and save it from polymyxin and let the polymyxin do the job and becoming this complex. And this is another example of silica modeling as we do docking design. It's done, as Dr. Farber did mention, without math, there's no past, no present, no future. So math is the main part of, and mathematical modeling of this, our functionality. And this is just one of the more sophisticated, more details. You can see it's very complex looking, but actually has their own explanation behind it. And it's silica modeling, and where we see pharmaceutical composition, which is composition which is protecting the kidneys against polymyxin. It doesn't let polymyxin to damage the kidneys, and it's right there. And nephroprotective agent, we call it NGL022. It has now our discovery the synergistic combinations. NGL022 has a way higher affinity to the same receptor that polymyxin loves to go to, not anymore. And it brings the results back to what we need to, what we actually try to come up with our research. So TRIZ and NGL022, that's our product and proof of concept. So as we mentioned before, we tried thousands of FDA generic drugs, 246 substances selected, out of which 2 substances synergistically combined. Only in combination, they work great as a nephroprotectors, and then don't let the polymyxin cause the problems. But, and this is properties, it's component of 2 groups. It's classes actually, sorry, 2 big, huge classes. And NGL02 has a higher affinity to megalin receptor rather than polymyxin, has minimal spectrum side effects. To begin with, it already has no side effects to the body, neither to megalin, and it does not affect polymyxin antibacterial activity. That's exactly what we planned and what we come up with. And here's it confirms by animal studies, in vivo studies. This is the markers. They based on the, it's a urine test that's done on the rats, and these biomarkers of the kidney disease. When we inject to the body, this is only polymyxin here, only, and this is polymyxin with our nephroprotectors. On the NOIGEL, this the blue one, shows on the startup when you inject those two medication and our composition, and the polymyxin and our composition. You see, he's trying to get to the kidney. Kidney start slightly not feeling great, and after therapeutic time, which is something needs to be up to week 2, 3 weeks treatment for those type illnesses, we don't have a side effects because our composition already blocked the kidney's damage from polymyxin, plus it doesn't have any toxic effect, neither on the kidney, neither on the body. But here we see just polymyxin makes tremendous damage to the kidneys based on excretion of this biomarker. This is what we had done afterwards. We even examined, it's called pathology reports. We have to, based on FDA requirements on the kidneys level, when we basically give that protocol, after the protocol of therapy, we had control group right here, only medication, and you see how much its accumulation of polymyxin grows exponentially in the kidneys until it could damage the kidneys and animal dies. And here you see slightly elevation, it still gets there, but not as much to cause major damage to the kidneys. It's great protection of the kidneys, and even on slices, pathological slices, it's confirmed. And we studied this for 18-month period, more than a year in different series. And we have a great data on this. And this is just combinations, 2 slides together, just for quick presentation on those. One of the toxicity check on the urine, and that's for the kidney pathology reports. Here we go. So we come up with solution. We got medication we need to treat our people, the humans, the patients. We got the megalin, which polymyxin loves to attach to, and they can't because we have a protector now. And since we're getting with protection, kidney is doing well, no nephrotoxicity, and we have polymyxin busy with their own job, what he's supposed to do without causing toxicity to the kidneys or to the further to the body. And this is just a little diagram here showing as polymyxin nephroprotectors without our nephroprotectors. That's the results we get currently. We cannot treat them as long as we need to. We cannot give them enough amount of polymyxin, or that important medication for the sick person, and insufficient treatment. Either person dies or survives with kidney damages, or it's some of the lucky patients who could just go through it. That's the reality. And here, the treatment with our nephroprotectors, that's our predictive factors again. We only done in vivo studies so far. And if we use our polymyxin with nephroprotection, there will be mild or no nephrotoxicity. If someone needs more than a week or 2 or 3 therapy for all kinds of diseases, there's plenty of disease needs up to even a month, like osteomyelitis, certain very deep tissue skin infection, gram-negative, and for survival, they need longer therapy. They could receive that efficiently. Where there's a market opportunity, of course, again, it's a problem currently. As we said, multi-drug resistance, based on my second or third slide, is growing. it became dangerous right now, and amount of money spent in pharma industry and healthcare industry and lives what we're losing because of that, it's tremendous. Well, we have a next step, of course. We have plans right now to work with nephroprotectors, which are generic. They use for different purpose approved, but it's called repurposing medications. So we use them for different, for this purpose right now. And we could work and increase their needs in the medical industry, healthcare industry. We're also looking for, of course, for VC groups in life science investors, so we could bring this to the next level clinical trials to use for the people and in the hospitals. And we open for discussions for collaboration. And our next study, as a matter of fact, right now we're working on aminoglycosides. It's big. It's one of the big needs in use right now. A group of antibiotics, and also having similar problem of nephrotoxicity. So we can revive them back to the market and use them for people who in need for the therapies. That's just quick our collaboration, what we're looking for. And absolutely, as Dr. Farber mentioned, we wouldn't come up to discussion unless we have intellectual property behind us and publications of our team and our companies. This is some of our pipelines and very few, but here's some much more. Dr. Farber did mention about them on his first presentation. And here we are, it's a team of the NOIGEL and TRIZ Biopharma company. And basically, this is the entire topic and about polymyxin and nephroprotection.